Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing IIT JAM question paper for the subject Biotechnology conducted in the year 2017. I have answered MSQ, MCQ and numerical aptitude test, all from biotechnology based questions. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of IIT JAM, you can go to my channel Omni Bio Entrance, select the playlist. In the playlist, select for IIT JAM and you will get it there. Coming to the first question. The antigen binding site of an antibody is present. It is present between at the variable region. Between the variable region of the light chain and that of the heavy chain. Then next one, which of the following is not involved in eukaryotic translation? Ribosome is involved, spliceosome is not involved. Spliceosome is the complex assembly of RNA and protein molecules. It removes introns from the mRNA, joins the exons and produces a mature mRNA. That is spliceosome. It is not involved in translation. It is in the transcription process. mRNA is involved in translation as well as tRNA. So the answer here is option B. Which of the following statements is correct? Gram-negative bacteria are colored purple after gram staining. They are colored pink or red after gram staining. Gram-positive is the one that retains the crystal violet purple color, and gram-negative do not retain the crystal violet dye. Gram-negative bacteria, they have thin peptidoglycan layer. So this allows for the crystal violet dye to be washed away during the decolorization step, and the safranin counter stain that you add later stains the bacteria red. This is what is happening in gram-negative bacteria. So they are not colored purple, they are pink or red. Then gram-negative bacteria are commonly more resistant to antibiotics than gram-positive bacteria. That is correct. Now the reasons for this resistance is the outer membrane having lipopolysaccharide, which is a barrier to many antibiotics. Then they have got porine channels. These porine channels, they will regulate the entry of antibiotics. Then there are efflux pumps that are also expel antibiotics from the cell. Then they have got beta-lactamase enzymes that break down beta-lactam antibiotics. So that is the reason why gram-negative bacteria are more resistant to antibiotic than gram-positive bacteria. Then next one, C. Gram-negative bacterial cell wall consists of thick layer of peptidoglycan. No. Gram-negative bacteria have thin layer of peptidoglycan layer. Then D. Cell wall of gram-negative bacteria do not contain an outer membrane. They have an outer membrane containing polysaccharides. So B is the answer. Then next one. The role of enzyme E synthesized by phage X174 during host infection is to block peptidoglycan synthesis. Answer is option A. Then next one. The result of electrophoretic separation of a mixture of amino acid X, Y, Z at pH 5 is represented as. So at pH 5, these are the X, Y, Z separation they have mentioned. Then given the isoelectric point of X, Y, Z. So they have given isoelectric point also. Here the basic principle is there is a relationship between pH and the isoelectric point, which is used for electrophoretic separation, isoelectric focusing. So pH is less than Pi, the net charge will be positive for that particular amino acid. If pH is equal to Pi, then the net charge is 0, it is at the Switter ion. Then if pH is greater than Pi, the net charge is negative. So if it is positive, it will move towards the cathode, and if it is negative, it will move towards the anode. These points you have to remember. Now coming to the question. for X we will see where it will be. X is having a pI of 9.87 at pH 5. So pH is less than the pI. So it will have a net positive charge and it will move towards the cathode. So X will come here at the negative side. That is there only in option A. Okay, we will see the others also. Okay, D is also having X at negative position. Okay, we will see. So B and C can be not taken into consideration. Then Y is having a pI of 3.22. Now 3.22, that is great, pH is 5. So 5 is greater than 3.22. So pH is greater than pI. It will have a negative charge. So it will move towards the anode. So Y will be at the positive side. That is here. Here you will see Y. Then next one. PI is 5.43 for Z and pH is 5. So it is somewhat 5.43 is approximately equal to 5. So the net pH is equal to PI and the net charge will be 0. So it will be in the center. So D can be eliminated. D they have given Z towards the negative charge, which is not correct. So A is the answer. Z will be in the center, X will be towards the negative charge, Y will be towards the positive charge and Z will be in the center. So the answer here is option A. Then next one. Which of the following is true of protein synthesis only in prokaryotes? Translation and transcription are coupled. Yes, that happens only in prokaryotes. It is not found in eukaryotes. In eukaryotes, transcription takes place within the nucleus. The mature RNA is coming out of the nucleus and translation takes place in the cytoplasm. Then B, the codon AUG codes for the start signal. That happens both in prokaryotes and eukaryotes. It is AUG. The tRNA anticodon can bind to 2 or more different codons. That is also correct based on Wobble hypothesis. As per Wobble hypothesis, single tRNA anticodon can recognize multiple codons. That happens, and Wobble hypothesis is true both in prokaryotes and eukaryotes. So C is also happening both in prokaryotes and eukaryotes. Then next one, the functional ribosome contains 2 subunits constructed of protein and RNA. Yes, there are 2 subunits for the ribosome, large subunit and small subunit. Even in prokaryotes, you can see 2 subunits for the ribosome. So D is there in both prokaryotes and eukaryotes. The question here is only in prokaryotes, so A is the answer. Then next one, match the entries in group 1 with that in group 2. Group 1, phytase. Phytase, it improves mineral availability. Iron availability is increased by phytase. So P4. So C and D can be eliminated. We will see A and B. Then silanase. Silanase is paper and pulp processing, Q1. Okay, in both it is Q1. Lacase. Lacase is delignification, R2. R2 is there in option B only. So B must be the correct answer. We will see. S also we will see. Bromelain. Bromelain is gluten complex reduction, S3. So answer here is option B. Then next one. In aldol cleavage of glucose 6-phosphate occurs in glycolysis, it will result in. So if aldol cleavage of glucose phosphate results, then it will give rise to 2 products. One is glyceraldehyde 3-phosphate and the other is erythrose 4-phosphate. Glyceraldehyde is a 3-carbon compound, erythrose is a 4-carbon compound. So it will produce carbon chain length of unequal length. So products of unequal carbon chain length will be produced. Answer is option B. Then the natural geographical distribution of kangaroos is limited to the Australian continent because Abiotic factors determine distribution. Not only abiotic, certain biotic factors also determine distribution in some cases. Then dispersal is limited by accessibility to other continents. That is correct for kangaroo, because during continental isolation, Australia got separated from other continents and this hindered kangaroo migration. Also, the surrounding oceans, they prevented kangaroos migration to other continents. So B is correct. Then kangaroos have not selected habitats in other continents. That is also not correct. Predators limit the distribution in other continents. That is also not correct. Then which of the following is not an example of adaptive defense mechanism against predation? So bright colors of bird pollinated flowers, that is the answer, because it is not a defense mechanism. It is an example of attractant, not a defense mechanism. It is for attraction. So it serves to attract pollinators. It signals nectar availability as well as enhance the pollination efficiency. So it is not a defense mechanism. It is an example of attractant. Then match the following. Group 1 to group 2. Nucleolus. Nucleolus is RNA synthesis. Okay, P4. Okay, C and D can be eliminated. We will see A and B. P4. Then spherosomes. Lipid storage. Q1. Q1 is there in B only, so B must be the answer. We will see R and S also. Peroxisomes. Peroxisome is breakdown of fatty acid. Yes. R2. Then plasmodesmata is transport of macromolecules. S3. So the answer here is option B. Then next one. The nitrogenase of diazotropes. So nitrogenase is an enzyme responsible for nitrogen fixation in diazotropes. So it is a complex of iron protein and molybdenum iron protein and uses 16 ATP to reduce 1 nitrogen. That is the correct answer. That is option C. Then next one. During eukaryotic cell division, the amount of DNA doubles. So amount of DNA doubles at the S phase. S phase comes between G1 and G2 phases. So prophase and anaphase of mitosis not correct. Prophase 1 and prophase 2 of meiosis no. Between G1 and G2 phases of the cell cycle, yes. Answer is option C. During M phase of the cell cycle, not at the mitotic phase. So that is also not correct. Then next one. The correct sequence of the following events in the human female reproductive cycle is. Okay. So first will be secretion of follicle stimulating hormone, FSH. Then growth of follicles and oogenesis, that is 3. Then comes sudden increase in the level of luteinizing hormone, 5. Then ovulation take place, 6. And then growth of corpus luteum, that is 2. So the answer here is option C, 1, 3, 5, 6 and 2. Now coming to MSQ questions. The cells involved in allergic reaction and contain surface receptors for IgE antibodies are and histamine are. So surface receptors for IgE antibodies as well as histamine, they both are present in basophil as well as in mast cells. They both are involved in allergic reactions. So A and B is the answer. Then next one. Which of the following is incorrect in the regulation of tryptophan operon? They have asked for the incorrect statement. It is an example of negatively controlled repressible operon. That is correct. The amino acid tryptophan inactivates the repressor. No. The amino acid tryptophan activates the repressor in the tryptophan operon. Repressor is the one that binds to the operator. We know that. And upon binding, it will block the RNA polymerase from binding to the promoter region. So what will happen in case of tryptophan operon is tryptophan will bind to the repressor. This tryptophan repressor complex, they will bind to the operator and block RNA polymerase from binding, thereby preventing the operon structural genes from synthesis. So B is incorrect statement. Okay, we will see others also. The amino acid tryptophan induces the operon. No, the amino acid tryptophan represses the operon. So C is also an incorrect statement. The repressor binds to the operator in the presence of amino acid tryptophan. That's also correct. Then next one. Which of the following organs are correctly paired with their function? Large intestine, protein, digestion. That is not correct. Large intestine is involved in water reabsorption, electrolyte balance, all these things. So large intestine is not involved in protein digestion. Protein digestion, it mainly occurs in the stomach and small intestine. Then oral cavity starch digestion. Yes, you have got the enzyme for starch digestion in the oral cavity. B is correct. Pancreas bile production. No, bile production is by liver and the gallbladder. Pancreas, it has got two functions. One is exocrine and the other is endocrine function. Exocrine function produces digestive enzymes and endocrine function produces hormones like insulin, glucagon and somatostatin. That is pancreas function. Small intestine fat digestion. Yes, that is also correct. So B and D are correct. Then next one. The delta G0 for homolactic fermentation converting glucose to lactate is minus 196 kJ per mole. If delta G0 dash for the formation of ATP is plus 30.5 kJ per mole, then which are the correct statements? So delta G for glucose to lactose they have given. Delta G0 for ATP formation from ADP and PI is also they have given. So this is energy required to form one molecule of ATP. Now if you see the equation, glucose plus 2 ADP plus 2 PI give 2 lactate plus 2 ATP plus 2 H2O. So number of ATP molecule produced per glucose molecule is 2. So total energy conserved in ATP is 2 into 30.5, that is 61 kJ per mole, or you need 61 kJ per mole for the production of 2 ATP molecules. Now you have to see energy efficiency. Energy efficiency is energy conserved in ATP by total energy released into 100. So energy conserved in ATP is 61. Total energy released is they have given for fermentation converting glucose to lactate is minus 196 kJ per mole. So 196 into 100, that will give you approximately 31%. The remaining out of 31, that is 69%, 100 minus 31 is 69. That energy will be released as heat. Now see the options. Homolactic fermentation is 31% energy efficient. Yes. Here if you see this energy efficiency we have calculated as 31%. So A is correct. The efficiency of energy conservation, it is not conservation, is 69%. Here energy is released as heat. 69% is released as heat. So it is not conservation. So B is not correct. The energy stored in the form of ATP is 31%. Yes. That is also correct. The process result in the loss of 31% of energy. No, it is not loss of 31%. It is loss of 69% of the energy. So A and C are the right answers. Then bacterial plasmid genes of non-chromosomal origin are associated with. So plasmid genes, they are mainly for resistance to various antibiotics, toxic materials, toxins, etc. Okay, now we will see the options. Providing resistance against antibacterial agents. Yes, that's correct. The degradation of toxic material. Yes. The production of certain toxin. Of course. The transfer of genetic material from one cell to another cell. Yes. So A, B, C, D, all four are correct. Then comes numerical answer type questions. So first one, antigen antibody interactions is shown by the following scheme. AB plus AG giving K1, AB AG dissociates by K minus 1 giving AB plus AG, where AB is the antibody, AG is the antigen. AB-AG is antigen-antibody complex. The values of K1 and K minus 1, 5 into 10 raised to minus 5 and 2 into 10 raised to minus 7 respectively. The dissociation for complex for constant for the complex is. So KD is equal to K minus 1 by K1. So 2 into 10 raised to minus 7 by 5 into 10 raised to minus 5. You get 4 into 10 raised to minus 3 micromolar divided by 10 raised to 3 for conversion. That is 4 in nanomolar. Answer is option. Options are not given. 4 nanomolar. Then next one. The population of a bacterial culture increases from 1000 to 1 billion in 5 years. In 5 hours. The doubling time of the culture correct to 1 decimal place is in minutes. So initial population is 1000, that is 10 raised to 3 or N0. And 1 billion is the final population, that is 10 raised to 9 NT. Time is 5 hours, or we can convert it into 300 minutes because the final this thing is in minutes. So doubling time is given by the formula T by 3.3 log N by N0, or T we have it as 300 minutes divided by 3.3 log 10 raised to 9 by 10 raised to 3, that is 300 by 3.3 into 6, that is around 15 minutes. Then next one. Km and Vmax of lactate dehydrogenase for conversion of pyruvate to lactate are 1 millimolar and 5 nanomolar per second respectively. At 0.25 millimolar pyruvate, the velocity of the reaction catalyzed by lactate dehydrogenase is. So Km and Vmax for the enzyme lactate dehydrogenase, which converts pyruvate to lactate. So pyruvate is the substrate, lactate is the product. So Vmax is 5 and Km is 1 millimolar, that is 1000 micromolar. Now they have given substrate concentration, that is of pyruvate concentration, that is 0.25 millimolar. So 0.25 millimolar, convert it into micromolar, 250 micromolar. The velocity of the reaction catalyzed by, so velocity is given by Michaelis-Menten equation. V is equal to Vmax concentration of S by Km plus concentration of S, or V is equal to, substitute the values, 5 into 250 by 1000 plus 250, that will give you 1 nanomoles molar per second. Answer is 1. Now next one. A linear DNA contains restriction sites for EcoR1 and 3 restriction sites for BAMH1. The number of fragments that will be generated after digestion with EcoR1. So BAMH1 you have to forget. They have asked only for EcoR1. So number of fragments will be number of restriction sites plus 1. How many restriction sites for EcoR1? 5. So 5 plus 1, 6. Answer is 6. Then next one. The standard oxidation potential for oxidation of NaDH and water are 0.315 volt and minus 0.815 volt respectively. The standard free energy for oxidation of 1 mole of NaDH by oxygen under standard conditions, correct to 1 decimal place, is dash kilojoules. Faraday constant they have given. That is 96500 calories per mole. You have to convert it into kilojoules. That will give you 96.5 kilojoule per mole. So one conversion is over. Now the formula that you have to use is delta G0 is equal to minus NFE cell. E cell is E oxidation minus E reduction. So E oxidation is E0 NaDH and reduction is E0H2O. Substitute the values and you will get it as plus 1.130 volt. Now substitute it in the formula. Minus NFE cell. N is equal to 2. So minus 2, F value you know, E cell value also you know, you will get it as minus 218.09 kilojoule per mole. Then next one. Km and Vmax of an enzyme are 4 millimolar and 0.1 millimolar per hour respectively. In the presence of 1.5 millimolar inhibitor, the Km and Vmax of the enzyme are 6 and 0.1. The value of inhibition constant. So here if you see, you can see that the Vmax after addition of the inhibitor has not changed. It is 0.1 in the absence of inhibitor and 0.1 in the presence of inhibitor. So it is an example of competitive inhibition, whereas Km value has increased. So it shows competitive inhibition. So you have to use the formula for when competitive inhibitor is present for Km dash. So Km dash is equal to Km 1 plus I by Ki. You have to find the value of Ki. Substitute the values. This is the calculation for Ki and you will get it as 3 millimolar. Then next one. The relationship between log molecular weight in kilodalton of a mixture of protein standards and their retention factors obtained from native pages. Log 10 molecular weight minus 2 RF plus 3. If the measured retention factor for a protein with 180 amino acid is 0.5. So RF value for 180 amino acid is 0.5. The number of identical monomers in the protein. So what they have given in the question is, they have given an formula for molecular weight calculation based on the RF value. Here they have given for a protein RF value of 0.5 for a protein of 180 amino acid. Now 180 amino acid, it represents the monomer. Not you have to identify the number of identical monomers in the protein. Okay. So number of monomers is given by the formula N into molecular weight of the monomer. You have to calculate the molecular weight of the monomer. For that, you should know the molecular weight of the protein. That you can get from this formula. So log 10 molecular weight of protein is equal to minus 2 RF plus 3. RF value is 0.5. So minus 2.5 plus 3, that will give you molecular weight of 100. Antilog of 2 is 100. It is log to the base 10. So molecular weight is 100. Now the protein has 180 amino acid and the molecular weight of one amino acid is 110 daltons. So the molecular weight of the monomer, that will be 180 amino acid into 110, that will be 19.8 kilodaltons. Now number of monomers will be n into molecular weight of the monomer. N you have to calculate, that will give you the number of monomers in the protein is equal to molecular weight of the protein. So n into 19.8 is equal to 100, n is equal to 5. So the answer is 5. Then next one. In bacteria, a ribosome synthesizes a protein containing 300 amino acids from mRNA in 20 seconds. Okay. The average lifetime of mRNA is 2 minutes, that is 120 seconds. Convert. When you see two different units, convert. See the question, what they have asked, whether it is in minutes or seconds. So based according to that you convert. So average lifetime of mRNA is 2 minutes, that is 120 seconds. The number of ribosomes that can translate a single mRNA containing 1350 nucleotide is. So you have got an mRNA of 1350 nucleotides whose average lifetime is 120 seconds. So how many ribosomes are required to translate that particular mRNA? Now they have given us the data, that is in 20 seconds ribosomes can synthesize 300 amino acids. Okay, single ribosome, it can synthesize 300 amino acids, or I can say it can translate 300 into 3, that is 900 nucleotides. 3 is our codon, triplet codon. So can 300 into 3, 900 nucleotides can be translated. Here it is 300 amino acids. So convert it into nucleotides, 900 nucleotides. So in 2 minutes, that is 120 seconds, it can translate 900 into 120 by 2, that is 5400 nucleotides. Now our mRNA, it is only 1350 nucleotides. So how many ribosomes will be required that can translate this mRNA is divide. 5400 divided by 1350, that is equal to 4. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year questions of IIT JAM Biotechnology, you can go to my channel Omni Bio Entrance. Select playlist. In the playlist select IIT JAM and you will get the question paper solved there. Thank you.